So right now we're going to discuss tetrad analysis where there are two genes that are unlinked. Right? And so the basic gist of this, as I said, you have to do what is called a haploid mating. You take these single haploid strains that have a genotype that you know, for example, and let's just say, for example, that this particular haploid strain that you have, which is 1N, has this particular genotype. Let's say it's Lu plus and Met plus. And you make that with another strain that's haploid 1N, and it's Lu minus and Met minus. Okay. So, one way of representing that is, basically on these markers residing on chromosomes. And what we're going to do is we're going to suppose that they're unlinked. Okay, so that means that these markers are on different chromosomes. And so we could draw sort of a configuration like this, whereby you have a chromosome which we'll label in blue, all right, and a chromosome which we'll label in red, all right. This sort of top pair, all right, would come from the first haploid strain. And then this sort of second bottom pair would come from the second haploid strain, like I was just talking about. Now, with regard to those two, all right, the first haploid strain was, well, I can't remember, but let's just say it was Lu plus, and it was Met minus. Let's just, I can't remember, so let's suppose it's that. And then let's suppose the second haploid strain would be Lu minus and Met plus, all right? So you've made a diploid and you're about ready to undergo meiosis. And so how does meiosis proceed? Well, of course, the first thing that you do is you replicate your chromosomes. And so you're going to make chromosomes that look like this. All right, with the sister chromatids. Right? And your other set of chromosomes that look like this. All right. And so this would then be Lu plus, Lu minus, Lu plus, right? Sisters are identical, right? Okay. And then Lu minus, Lu minus. Okay. And then this would be Met minus, Met minus, Met plus, and Met plus. Okay. Now, what's important here is that if you undergo meiosis in the first phase of meiosis and meiosis 1, the homologs will segregate away from each other. And so what that will do is produce a particular arrangement in meiosis. So now I'm going to erase this top part here and sort of show how this will sort of set up in meiosis. All right, so the top part will segregate in the first meiotic segregation. So what you end up with now after meiosis 1, all right, is going to be sort of like, you can imagine, two nuclei, essentially. One of which has these two genotypes, and the other one which has these two genotypes. So it will just simply be showing the chromosomes. This and like this. And the genotypes then showing the first two segregating to this part and the second two segregating into this part. The Lu plus, and then Met minus, Met minus. Met minus, Met minus. And then the opposite, of course, on this other side. So Lu minus, Met plus, Met plus. Okay. Now, obviously, if you then undergo the second round of meiosis, this will segregate into 4 haploid products now inside the large tetrad. Both of these would have the same genotype, right? And then both of these would have the same genotype. So I'm just going to draw genotypes now as the haploid products now. So this would be Lu plus Met minus, and then the other one would be Lu plus Met minus met plus. Now what's important here is basically there's two genotypes in this tetrad, right? There's one of the parentals and then the other parental. So the fact that there's two types means it's a ditype, and the fact that both of them are parentals means it's a parental ditype, right? Now one thing that's important to understand is how often do you get this conformation versus the other conformation in which Lu plus is now with Met plus. So these ones are basically switched, right? Now remember something, right? This particular arrangement of this set of chromosomes and this set of chromosomes was established in meiosis 1, right? But it's equally likely that this one could switch with that one, right? So it's equally likely that these two segregate together or these two segregate together. So after meiosis 1, the other combination in which you have, for example, let's just change them here, this conformation, Met plus with the Lu plus, and the Met minus with the Lu minus, that's as equally likely, just the way that the chromosomes line up in meiosis. And when you have this particular conformation, Lu plus with Met plus, Lu minus with Met minus, so I'm going to change these, Lu plus, Met plus, Lu plus, Met plus, Lu minus, Met minus, Lu minus, Met minus, alright? That's as equally likely, but that's not the parental genotypes that came in from that haploid mating, because here you see that both of these are plus, right? Likewise, both of these are plus, and both of these are minus, and both of these are minus. So, the point I'm trying to make here, what is this particular tetrad? Well, this would be called a non-parental ditype, because there's two kinds, 1, 2, this pair, and this pair, right? And they're both the non-parental kind. So we would call this a non-parental ditype. And the critical thing I'm trying to make, the critical point I'm trying to make here, is because of the way that the chromosomes segregate independently, just independent segregation, the numbers of non-parental ditypes among all the tetrads that you analyze would basically be equal to the numbers of parental ditypes. And that's what tells you that genes are unlinked doing tetrad analysis.